right, great. All right, everybody. Yeah, super excited today for our first meeting of 2026. We've got a really exciting startup, Manifold Bio, who's coming to present today. So we've got Erik Swanson and Mike Nichols. So just very brief intro. Erik did his PhD. I'm sorry,
Undergrad.
Erik did his undergrad here in Boston, actually studying black holes, and then transitioned over to protein design. While Mike did his PhD at Emory before moving up to Boston. Both of these guys joined Manifold right out of school and been doing some really kick-ass stuff. So, guys, take it away.
Yeah, thanks for the introduction. Just to start with, I want to thank really Chris and Nick and Sergey for organizing this thing. We've been coming here for a few years now, and you know, we've learned so much from all of the speakers who've presented here, and so we really wanted to give back to the community here by talking as well. We certainly wouldn't have gotten as far as we have without you know, all of the great speakers who have been coming through this venue. So with that, I'll give it over to Erik to sort of introduce mBER.
Yeah, thanks, Mike. So today we're going to be sharing with you mBER, which is our de novo protein design system. The preprint came out back in September, so folks might be familiar with it. But you know, the tagline for mBER really is controllable de novo antibody design with million scale experimental screening. So not only are we doing some new design, but we have this massive experimental pipeline, and we think that's a really exciting part of the paper here to present for you. There we go. All right. So to outline the sort of structure of the talk here, I'm going to start with a background that probably most people will be familiar with, which is the story of AI and protein design, from protein folding to binder design, and then to our stage here, which is antibody design. We're gonna go from that to talking about the experience of developing mBER. What were the key insights that led us to our method and why we think it works. And then we're going to really dive into the experiment. Like I said, this is a really exciting part of the paper. It is, to our knowledge, the largest wet lab validation, largest design campaign and wet lab validation of de novo I think binder design in general, not even limited to antibodies. And then from there, we're gonna talk about why are we doing this at all at Manifold? Why are we designing million scale libraries? What is it all for? So without further ado, let's talk about this story again. We'll go a little quick through here because I think people will be familiar, but it's worth touching up on. So we can go back to July of 2021. For many decades, the protein folding task was maybe the biggest problem in the field. I don't think it'd be strange to say. We're really interested in this question of, given a protein sequence, a string of amino acids, can we predict the 3D physical structure of that protein? AlphaFold, famously, quote unquote, solved this problem. Racing past all of the other competing models and achieving really, really high performance on the public benchmarks CASP. So AlphaFold originally solved the problem of monomer folding. They very quick after that gave out their approach to the problem of multimer folding, which we think is much more interesting. We're not only interested in how proteins sort of exist on their own, but how they interact with other proteins, especially when we're talking about doing experiments in living systems. And of course, AlphaFold, along with some protein design work by David Baker, won the Nobel Prize in Chemistry in 2024. So we talk about going beyond folding to protein design. We're not only interested in mapping from sequence to structure, but also going all sorts of different directions. So I've outlined here a couple common patterns for protein design. Some models will just generate structure unconditionally or conditionally. Some folks are generating sequence conditionally or unconditionally. You can map backwards from structure to sequence. It's like an inverse folding task. And of course, folding, you can go the other way. For us, we're particularly interested in the subproblem of protein binder design. So we say, given some known sequence and structure, can we design a new protein that will bind to that physically, hopefully have some sort of effect? We're very interested in real effects like therapeutically in vivo, but of course you can measure these things in vitro experimentally. So when we look at the history of protein binder design, there's a lot more here that I'm not touching on, but these are the the key methods that have been really influential to us. So in December 2022, R-Diffusion was released. R-Diffusion was, I think, really the first really well publicized mainstream method for generating de novo protein structures. You can argue different ways about that. But anyway, R-Diffusion generates a protein structure without a sequence. There's various conditioning that you can apply to it. You can, generate structures to bind other structures, other structure-based stuff as well. Protein MPNN is a separate model, which does the inverse folding problem we were talking about. So once you have a structure with no sequence, if you want to actually make that thing exist physically, you do need a sequence. So oftentimes what people will do is they'll generate the structure with R-Diffusion, generate the sequence to that structure with protein MPNN, and then refold it without fold 2 to see, okay, does the sequence fold into the structure I wanted? And then hopefully, it'll work if I order it in the lab, right? A couple years later, in October 2024, we saw the release of Bindcraft, which builds on Colab design, which was largely created by Sergey, I believe, and probably some folks from his lab, but I think largely Sergey. Bindcraft was really exciting to us because it was this whole other method, and specifically it came with experimental validation. So what Bindcraft showed is that you actually don't need all these fancy diffusion models. You can just take, AlphaFold, which is a really great folding model with a confidence metric. So not only does AlphaFold give you a structure, but it tells you, how confident am I that this is a good structure, that these are, correct atomic placements, right? And if you just directly optimize on that confidence label, then you can get really, really high quality binders that will, express and validate in the lab. So we saw this and we were like, whoa, that's really cool. It's really cool, but both R-Diffusion and Bindcraft are currently, I think still, at least in their initial versions, limited to generating what we generally term mini binders. So these are they're binders, but other than that, there's not a lot of conditioning. There's some examples in the top right here in this GIF. They're the binders in blue. They often have this alpha helical structure. We think it makes it pretty easy to bind in secondary structure interaction ways, surface complementarity stuff. But in terms of the design problems we're interested in, mini binders are great tool molecules. They work in the lab. But for therapeutics, we would really like to be able to design antibodies. And there's a lot of reason for this. I won't go into all of them, but largely it's because antibodies have a long history of clinical validation. So, if you go to a pharma company and you say, I have a new drug for you, and you say, this drug, it looks nothing like anything you've ever tested, then they might say, maybe not. But if you give them an antibody, they'll say, great, we've done that a lot. We'll do the standard suite and hopefully it'll work out. Antibodies, unlike mini binders, are thought to be quite hard to design. Some of the main reasons are, you can see in the structure to the right here, this is a single domain antibody. The secondary structure here is largely beta sheeted, which is well known to be a bit harder to design. And the binding interaction happens largely through these disordered loopy regions. These are the CDRs, or complementarity determining regions. AlphaFold is not excellent at folding these. And in general, it's, I think very much an unsolved problem for understanding all the possible ways in which disordered regions can bind. Nonetheless, we were very interested in antibody design. And when we saw Bindcraft come out, we thought, well, AlphaFold's kind of bad at folding antibodies, but it's not that bad. There is some success, right? So if we could just get AlphaFold to fold antibodies in a reasonable way and give us some high confidence results, maybe we would see some success. So what we did is we took the backbone of AlphaFold and Bindcraft, and we threw in a couple additions here. We grabbed some protein language models. Here we're naming ESM2. Some auxiliary folding models. We're using some from the Oxford Protein Informatics group. You might be familiar with Immune Builder or an Antibody Builder 2. And also we have a long history of our own as long a history as you can have of de novo binder design in our wet lab, which informed a lot of the decisions we made here. So we combined all these things into what we call mBER, Manifold Binder Engineering and Refinement. So how did we actually go about developing mBER? And to kick this off, I want to introduce a couple of what I think are some key ideas for binder design. The first one is, and it really is our key questions. Can you develop an evaluator or filter for in silico designs that has good translation to in vitro success. So for us, this often looks like, if you fold something with AlphaFold and it tells you it's very confident, we hope that thing will work in vitro. We've measured that a lot of times, and in fact, it does. There are lots of other filters that you could use, and I think it's still an open question to the field, which filters are best. And then the next question is, subject to such a filter, can you develop a generative design system that passes that filter? And I put in quotes here without cheating. And what I really mean here is like there's a lot of pathological ways that you can trick these filters with something that doesn't look at all like a protein. So if you make something that looks like a protein and it passes the filters and your filters are good, then you've probably hopefully done a decent job at binder design. Okay, so again, a little bit back in time here. As soon as ArcFusion came out, we were like, this is really cool. Let's try it out. What's going to happen? Manifold, our big advantage is that we have massive throughput for all of our wet lab experiments. So immediately we said, all right, let's just design like 100,000 of these things and test them. Like, do they work, right? So we did that. We designed against more than 600 distinct targets with ArcFusion. We used the standard pipeline here, ArcFusion designing of target structure, a binder structure, protein MPNN redesign of the binder sequence, fold with AlphaFold, order accordingly. And this is a plot. It's an old plot, so sorry it doesn't look that good. But it shows basically the distribution of binders across an IPTM range. IPTM being one of the sort of major confidence metrics from AlphaFold, which just says essentially, how high quality is the interface that I'm predicting, or how high quality do I think it is. And we show here the blue line is the success rate, which corresponds to the axis on the left. I know it's kind of hard to read. The point here is that as you get into IPTM ranges around like 0.7, 0.8 higher, you see success rate dramatically increase in the wet lab. This is in vitro success rate. So we were shocked. We were like, wow, you can actually get binders upwards of like 10% success rates in the lab. This was our first pass at it, and we were stoked. Yeah.
Quickly, how did you guys do the lab experiment, and what do you define as success?
That is a great question. We'll cover that with respect to the mBER paper because the experiment there looks a lot like pretty much all the experiments we run. Yep. So it'll come in the third part here.
On that effectiveness score, why does it drop when you get really high confidence?
So the red line is showing the distribution. Essentially, there's very, very few actual binders in that region, so it's just a sampling issue, I think. It looks smoother than it really is.
It could be some other things too. There are like some pathological things that pass at really, really high IPTM that we might just not have caught, but it's probably mostly a sampling problem.
That is another good point. If you see a really high IPTM, oftentimes that tells you something's wrong, actually, if it's too high.
Yeah. Cool. Also, in short, the assay here is just a bind, no bind assay. That's the short answer. So we say if we measure it binding, it's good. If we measure it binding, it's not. But again, we'll get into it more in a bit. All right. So going down the line here, when Bindcraft came out, we saw these results. This is a figure from their paper, basically showing, yes, they were able to generate these mini binders to a pretty diverse set of targets with like really, really high success rates. So some of these targets, you can see they got like, 6 out of 6 designs, maybe 6 out of 7, like basically 100% success rate subject to their filters for a lot of these targets, which was great for us because we kind of thought like, IPTM works for ArcFusion because ArcFusion is good. Actually, like maybe IPTM works because AlphaFold is good, right? Which we really was the lesson we took from Bindcraft. So then we asked this question like, yeah, how do we actually design antibodies, right? And why can't Bindcraft design antibodies? And I've liberally stolen some slides from Sergey again. But to understand the question here, you have to understand how AlphaFold predicts structures to begin with. And I think it's pretty well agreed upon that AlphaFold, there's a lot of complexity in the model, but before any of the folding actually happens, AlphaFold typically will take in either information from evolutionary information from multiple sequence alignments, MSAs, or templates, or both, where templates are previously known structures. And what this does, well, in the MSA example here, actually, it's kind of an interesting story. If you look back at evolutionary history of a set of closely related sequences, sometimes what you'll see is that a pair of positions, every time there's a mutation in one, there's also a mutation in the other. What this tells us is that there's some important coupling between these two residues, And more than likely, what that means is that those residues are actually structurally in contact. So Sergey's given a lot of great presentations on this, but we think that basically the MSA module is processing this data, turning it into a contact map for AlphaFold, so it can take a global guess at structure. It solves really like most of the problem of folding, other than kind of local secondary structure relaxation, some other things. But MSAs are quite annoying and expensive to compute. You often have to like query a really, really big database, especially if you're trying to do de novo design. It's maybe actually just not that useful. For antibodies especially, like the CDRs, actually, these are the parts that bind. They probably don't have a lot of evolutionary signal, because the way that CDRs sort of exist is your body sort of designs them on the fly to match whatever antigens you're going after. So we really didn't want to rely on MSAs, inefficient and maybe not so effective for our use case. But how do we get around it? I've alluded to this already, but with the template module. So AlphaFold is allowed, actually, if it doesn't get MSAs, or even if it does, to sort of cheat by looking at all known structures, say, here's some structures that have similar sequences to the thing that I'm folding. I'll just take the contact map or histogram from that structure, and I'll use that to help me, figure out what this should look like. So, this has been done by a few other groups, but we sort of co-opted the method here. We decided, what if we just hack the template module of AlphaFold by providing it some models from, pretty good antibody folding models. Again, these Oxford Protein Informatics group models are quite successful, at least at modeling the frameworks of the antibody, which are very important for keeping the structure and decent at the CDRs as well. But of course, it's not only a structure that defines an antibody. We also care about the sequence. Again, this will be familiar to people with antibodies experience. It might not be if you don't, but antibodies have these framework regions, which are very conserved. The sequence is the same. The structure is largely the same across, antibodies with the same germline. The CDRs are highly variable, but it's structured diversity. Like, it's not just random. There are certain amino acids that are preferred in each position in the CDR. So we thought, this would be important to optimize and make antibodies that actually looked and folded like antibodies, rather than letting AlphaFold run willy-nilly, design whatever it wants. So to take care of that, we introduced some biasing from ESM2, protein language model. We experimented with a few other language models as well. It turns out ESM2 is pretty good. And we essentially, we feed this bias term into the design. So every step when we pass a sequence through the model, we really, really strongly constrain the frameworks. We say, bias term is so strong here, you're not allowed to change anything. You're not able to change anything. And then we adjust the bias in the CDRs, sort of to our liking, depending on how much we want this sequence to look like a natural antibody sequence, according to a protein language model or not. So what do these, yes, please.
So I guess ESM2 has the same antibody sequence during training, or I guess I'm wondering, there's also like antibody specific language models.
There are, yeah.
So the question is, has ESM2 seen antibody sequences, and why didn't we use antibody specific language models?
Yeah. The short answer is we actually did in many cases. We also did some like internal fine tuning on antibody sequences. The truth of the matter actually is that like ESM produces, I don't know how many antibody sequences it's seen, but it does produce pretty good CDR distributions. And we just didn't see any improvements with our fine-tuned antibody models. It was kind of early in the development, so there could be more detailed analyses to run. But we figured, especially for keeping things open source and as understandable as possible, we'd use the, I think ESM2 is really the most common protein language model, and it seemed to work. So, yeah. Okay, here's a look at the actual effect of these two components in the design trajectory. So on the top left, you can see what happens when we provide a VHH template, but no real sequence prior. So here we're actually not even providing the frameworks. We're just asking AlphaFold to design something to a VHH template. It fails dramatically. The confidence here is actually the color scale as well. So you can see the red is AlphaFold's very unconfident about this. It's kind of a mess. On the top right, we're showing what happens when you provide just the sequence and the sequence prior, but no VHH template. AlphaFold does a bit of a better job. It's kind of antibody-like, but it's still quite low confidence, and there's a lot of disordered regions that shouldn't be there. In the bottom, of course, we provide both of them, and we get nice high confidence plausible VHH folds. Also note here, in case I didn't earlier, when we say antibody here, we are referring to single domain antibodies, VHH, not full antibodies. In theory, the method could apply to both, but we've worked mostly with VHHs internally. These animations, yes.
The question is, what happens to the confidence when the CDR3 gets long?
It is harder to design for sure. It is very target dependent. So there are some cases where you can actually get like a nice like long beta sheet where only the tip is disordered, and that works for certain binding modes. There are other cases where, like, if you look at a bunch of nanobody structures in complex, there are some really nice long loopy CDRs that form a really cool hydrogen bond network. That's a lot harder to do. We have a couple examples, actually, sadly not in the deck. Well, maybe there's one in the deck. It's harder to do. Yeah, usually AlphaFold is quite uncompetent with very long CDRs. Yeah, in practice, we have designed, I believe, up to, I want to say 15 residues in the CDR3, so pretty long. We have seen successes on that range. I wish I had a plot here for you for success rate across the range of CDR3 lengths, but I don't. So maybe in an update to a paper at some point. Cool. This is the same picture as before, but just the animation of the actual design trajectory. I'm not going to go too deep, by the way, into the actual trajectory. Bindcraft really covered that quite well. If people are interested, you can ask questions. But again, what we're doing here is that every single fold, we get a structure and a confidence here represented by red versus blue. And we just feed that through some loss terms to AlphaFold. We backpropagate to optimize the sequence with respect to those terms. All right, so in the top left here, you see that when we don't provide any sequence information, it's ugly. In the top right, when we do provide the sequence information, but, well, okay, I already went over it. Basically, top two are bad, bottom one is good. And the bottom is actually good for most of the trajectories. So, and this we see very often is like when we get successful designs, actually AlphaFold is quite confident about them throughout the whole design session, which is, we think, useful sort of like in the picture of almost like adiabatic cooling, where it's like each subsequent fold is not that far from the last set of sequences. Yes.
So are you starting with the template sequence as well, or are you starting from a random sequence that are arriving?
Yeah, so to be fair here, we actually are not starting with the framework sequence in the top left. So it is not the full set of ablations that you could run for the value of the biasing term here. This was the most dramatic one, which is why we included it. We found in early experiments that trajectories tended to converge better when we provided the CDR biasing. Yeah.
Can you explain what I'm seeing in this window PLDDT? I see a structure. You said the CDRs were 15 at most, but the thing that's varying is much longer than 15. And you also said the CDRs were disordered, but this looks very ordered. So I'm confused.
Sure. Yeah. So in the bottom section here is probably a little bit easier to think about in terms of CDRs. So the entire VHH sequence is approximately 120 residues long. That's shown on the horizontal axis here, position 0 through 120. On, I guess, the vertical axis, you can see basically these are 20 different amino acids, and where there's a blue block, that's the amino acids that's been chosen. At each step when we're folding, we're actually not always passing a real sequence. We're passing oftentimes a continuous representation of a sequence across many different amino acids. So what you can see, I hope the microphone will catch me if I go over here. Maybe I can motion with the with the mouse. Basically, the very stable regions here, these are the frameworks where we've strongly constrained the sequence. The mutating regions here are CDR1, CDR2, and CDR3. So you can see that CDR3 in this case is, I don't know, maybe 12 residues long. We also, our CDR1 and 2 definitions are slightly non-canonical. We include a little bit more of framework 3 in the design there. For VHH, it's especially important because they tend to bind on their side.
And just for a little bit more global orientation, there in the structures that you're looking at, there is a fixed structure that is the target that we are designing against. So that is not changing. We're not designing that. It sits blue because we're providing a template that AlphaFold is able to say, yes, this is how this thing should fold. And the thing that's wiggling around is the entire VHH, which includes both the frameworks, which are sort of well organized. They'll look like beta folds, beta sheet folds, and the CDRs, the complementarity determining regions, which will look like little disordered loops.
Exactly. Yeah, and the bits here that are lighter blue, this is less confidence. Those are the CDRs. So that's the sort of lack of confidence disorder.
The last thing, the heat map on the right is the PAE, or predicted aligned error matrix, where essentially the top left quadrant represents the target itself and its predicted aligned error, which remains blue in every case. The bottom right is the binder itself. So essentially how confident AlphaFold is that the binder itself is well folded. And the top right and bottom left quadrants are the confidence with which AlphaFold believes those two structures are placed with respect to each other. Yes.
I mean, the CR3 length is it's a hyperparameter, or do you have a way to sample that as well?
Yeah, in practice, what we've done typically is we have a set of frameworks and CDR lengths that we just most often use. So if you're going to run mBER, you'll actually just give it a full sequence and replace all the CDR residues that you want to design with like asterisks or star tokens. So in the middle of design, you can't change the length currently, but you can give it whatever you want at the start. We run a bunch of different trajectories with different frameworks and different CDR lengths. Great. Oh, sorry. Yeah, sorry.
So it looks like it's jumping between like two different poses. I'm guessing maybe that's like model 1, model 2, and outfold. Do you know which one? It kind of looks like it's settling on both solutions. Do we actually know which one is the right, or the solution that's actually?
That's a great question. I think it's the one where it's more aligned. I believe this is like PDL1, and most of the successes that we've seen, the final structures have been with the more aligned, not the like cross beta sheets, but this one.
Does this always happen? That it fluctuates? What I'm wondering, maybe model 1 or 2 has some weird bias towards the wrong structure or something.
It's a good question. We see it often, for sure.
We actually use 4 models, right? We use 4.
Yeah. We do a couple changes from. Yeah. Yeah, I mean, that's very possible. It'd be interesting to examine the trajectories in more detail. Unfortunately, the trajectories are like a lot of data, right? If you generate 1 million binders and each one of them has like 100 steps, then that's 100 million structures to analyze. It's not, it's a little difficult.
Yeah, you'll see coming up, but we're generating to so many targets, so many binders that we wish we could stare at these and learn from them. We think there is a lot to learn, but we don't have the human bandwidth to pay attention to every design.
Yes. I think another question.
Do you bias the epitope in any way? Like, why is it sampling only that part?
Yes. Yes, we do.
We'll get there.
Yeah, we'll get there in a moment, actually. Yeah, that's. In fact, we might as well get there now. This is the maybe like graphical abstract from mBER. This is the high level picture of how the pipeline works. It's actually quite simple, especially if you're familiar with tools like Bindcraft. And we make this claim, backprop plus conditioning plus bias is maybe all you need for protein design, right? Is that if we can actually, we know that AlphaFold Multimer now can design high scoring antibody binders. Maybe that's enough, right? Like if we can just get really high confidence folds, get things that look like antibodies, both in sequence and structure, maybe they will work in the lab. And sorry, I promised I'd answer the question here. One of the first steps in mBER, there are a lot of ways to do this, but typically what we do is we'll start with a full target, either from AlphaFold database or from the PDB, and we'll select usually actually one hotspot, but sometimes multiple. We have a truncation algorithm that basically tries to cut down to the set of important residues around that hotspot based on things like distance, chain breaks, and actually the PAE matrix in AlphaFold prediction cases. So that biases our selection in 2 ways. One way is that we actually usually just don't give the whole protein to mBER. That's great for efficiency and also making sure that it doesn't bind something that it shouldn't. There's also a loss term, sort of Bindcraft style, to bias towards the hotspots, although that's actually less effective, as you'll see, than the truncation. Great. Let's get into the experiment, which in short is all against all phage screening. So I'll pass it off to Mike.
Yeah, so phage is short for bacteriophage. It's just a little bacterial virus. And we use it because it's a simple way for us to sort of conjugate a DNA sequence that we can very easily read with next generation sequencing to the protein that is derived from that DNA. So if you think about what a phage looks like, what we're doing is we're essentially displaying the VHH on the tip of the phage, and then inside of that same phage will be the DNA phagement that gave rise to it. So what we can do is we can order very large oligo pools, from people who print DNA, clone them into a phage vector, and then raise very large libraries of these phage. We can then essentially just tile our target proteins, our proteins of interest, across a bunch of different wells, across many plates, flow that phage library over into each well and see what sticks. Then we just elute those bound phage and sequence it. And the result is essentially we can tie to every single DNA sequence its propensity to bind in every single well. The power here is that we're not just evaluating each binder to the target we designed it against, but rather we're evaluating every binder to every target simultaneously in a single experiment. So it's an all versus all assay, which gives us enormous statistical power. That phage can be somewhat noisy of an assay, but the huge number of sort of wells where we have multiple replicates of one on-design well, one target binder is intended to design, as well as hundreds of targets that the binder is not intended to design, give us the power to identify these things. And so also Eric and I have to thank Supriya Ravichandran, who does almost all of the wet lab work related to the paper here.
Single-handedly.
It's very impressive.
A lot of plates. Yeah.
How do you go from 120 targets to 70 million?
Yeah, so it's just so imagine I have a library with 600,000 unique DNA sequences in it, and now unique phage that all have so there's 600,000 designed VHHs, and then there's 24. I don't see the math. 120 target.
600K times 120.
It's 600,000 times 120 gives you 72 million measurements, most of which will be negative, right? Most of which will be binders. We're evaluating binding to a target that something was not designed against, but give us the ability to identify those positive binders by their negativeness. Yes.
For each pair, I'm getting some binary signal of binding or some continuous value.
It's a continuous value, so it winds up being an NGS, like a next generation sequencing continuous readout, which is very useful for us. Yes.
We'll get into the hit calling algorithm, off of that, turning that into a binary label in a bit. Yep.
And the 600K, is that independently synthesized? It's not combinatorial. It's like 600 independent designs that come out in models.
Yes, mostly we order oligo pools from Twist mostly.
Yeah, this is the point. So you have 600K for a target.
No, so it'll be 600K, and that'll be divided across the 120 targets. So on the order of 10,000 binders per target. Yes, exactly. So they're computationally designed for every target we are looking at all the different epitopes we're interested in, and we're designing thousands of binders and testing all of them. Yes, part of it is that we get it for free, right? We don't. It's all just one experiment, one big sequencing run. But the other part is it helps us discriminate. There's many reasons actually. We think it's very useful. One is that we actually do see off-design success rates that are lower than on-design, but help us benchmark and understand how well our design is really working. When you see if you design something to target A and it binds to target B, that gives you some sort of understanding of the baseline binding rate, right? Of sort of how good am I really doing at designing? I think people just assume that any amount of binding is good, but if your binding success rate is actually comparable to that off-design rate, then you actually don't have any lift, right? You're not actually really designing, you're just sampling from this library, and sometimes you get lucky, right? The other thing is specificity, we think is actually underappreciated in the field. We see lots of binders that bind to their target specifically, but also bind in a polyreactive way to even just 3 or 4 or 5 or 10 other targets among the 100. So they're not pan-reactive, but they are polyreactive. And so we think we're really jazzed about being able to see that and being able to learn about it and being able to predict it. Yes, exactly. So yeah, we're able to say these things are not just binders, they are specific binders. Yes.
How much understanding where it's are binding?
So we don't have epitope information in that way. Like all we can see is that we are binding to a target. Importantly, if we do have a significant amount of on-design success versus off-design success, we do feel that the extent to which that there is a delta there. Let's say it's 100-fold greater on-design success there's an off-design success. You can sort of crudely argue that gives you a 99% confidence that the structure you're looking at least contains some information, right? It's unlikely that you happen to get very successful binders and they're binding to an epitope that you didn't design them to, if they are indeed binding on target sort of more than they would off target. So we think we can sort of infer their epitope in that way, their structure. Yeah.
How many experiments are they in the works for transient binding? So more of an in vivo approach to understanding both where these bindings are happening and possible antibody interactions that don't last, that you wouldn't be able to catch with the phage assay.
So by transient, you mean like something with a very fast off rate, essentially? No, I don't think we've sort of. I think that's a hard problem to tackle, right?
Phage display is very bad.
Yeah. Okay. Then we're probably already catching them. Yes.
Some intuition of why 600K to 1. I mean, is it because that's the ratio to get a success? If you used 10K, would you just get no hits and use 6 million?
Yeah. We'll talk about success rates. 600K is really just chosen because that is what mostly that is the largest oligos library size you could order from Twist. So it's just practical purposes. Their DNA printer, I guess, runs up to 600,000 sequences. So it becomes computationally expensive to order more. And of course, the experiment does have some sort of limits on signal to noise ratios. Right. So as you increase the number of binders even more, the signal you're getting to every sequence, the amount of phage, the proportional amount of phage in every well decreases. Right. So in fact, actually, we've seen that if we decrease that number to 600K from 600K down to 100K or 10K, we do see like slightly higher hit rates, for instance, even if we're not changing anything, just because our assay becomes more sensitive. But by going to 600K, we sort of sacrifice a little bit of accuracy while maximizing the number of sequences we're evaluating. So in the end, we wind up with more hits. But of course, at some point that would flip and the experiment would become worthless. Yes, Chris.
It's all very exciting. So sorry to ask maybe some nitty-gritty questions. Maybe you'll get to this, but what fold over diversity did you guys sample your library? I'd love to learn a little bit more about like how reproducible the results are between wells.
Sorry, what do you mean by fold over diversity?
Well, you've got 600,000, right? So like how many phage particles do you put per well, right? Are you like oversampling your diversity about a 10-fold, 100-fold, 1,000-fold?
That's a great question. I don't know. The question was how many, how many phage particles are there per well? Sort of, so essentially, what is the ratio of the design, the complexity of the library, the number of unique designs in the library, and how many actual unique individual phage particles went into a well? It's certainly enough that we see replication. I don't know how much.
You oversample your library, you might suppress non-specific interactions or low affinity interactions. If you undersample, right, then you might not actually be getting super reproducible results.
Sure.
A little sparse. So
yes.
How do you pick what you sample?
Yeah, I think the answer is Supriya might be able to give you a more intelligible answer than I can, but I believe we sort of tried to maximize titer. And I think in this regime of 600K, we're far more likely to be sort of undersampling than oversampling for our wells.
The short sort of like data-driven answer is that we do 4 replicates per target binder interaction, and we actually our entire hit calling strategy is based on agreement across those replicates. So we just won't call something a hit if there's any disagreement in that measurement.
would say any disagreement.
Well, okay.
Okay. So just to give you a sense of what the in silico side of this looks like, we nominated 436 unique targets. So we didn't make this easy for ourselves. We essentially just went through the human proteome and nominated a bunch of the only real criteria were things that were human cell surface receptors, so things that would be exposed on the surfaces of cells. We nominated 436 of those and designed binders to as many epitopes on the surfaces of those proteins, the extracellular domains of those proteins, as we could. What you're seeing here is sort of all of the we get very variable design successes. So in gray is like early stopping. So we have early stopping criteria that if a trajectory looks like it's not going to be productive, it stops. And then the different colors represent IPTM thresholds of the final designs of 0.7, 0.7 to 0.8, et cetera. And ultimately what we think is successful or what we think will work are these things in sort of the 0.7 and above or even 0.8 above. So the point here is you do have to sample a lot to fill out a library like this full of binders that you think will actually work. And in fact, we're not actually giving every single one of these targets an equal shot. So we're just designing as much as we can in the timelines we're afforded. And so some targets are very highly sampled. There's going to be lots of binders to them with high IPTMs. And some targets were, for whatever reason, the design algorithm isn't able to, mBER is not able to generate high predicted success binders. But that's the library we move forward with. Yeah.
Is there anything different between the types that work well computational and the one that don't work well? Like if you look at the surface, are they more hydrophobic, less hydrophobic? You could give an analysis.
There is a lot more analysis to be done there. We are starting to look at that data or look at it in that way. We don't have anything we can share, unfortunately, in that way. We think there are a lot of failure modes that are actually fairly obvious. There's things that we're not considering here, for instance, like post-translational modifications, dimerization interfaces, these sorts of things are obvious failure modes of the model that we need to do a better job of accounting for. But yeah.
I guess I could take a follow-up question.
Yeah.
How did you make the 436 antigens in vitro?
So yes, that would be very impressive if we had made them all. We did not make them all. We made
145.
I think it's 120 or so, right? Or
There were 145 in the experiment. I think
okay.
we didn't make all of them.
We didn't. So we sourced them from a variety of sources. Some of them we made in-house. Some of them we sourced externally. And some of these, frankly, cannot be sourced. They are things like multipass membrane proteins that aren't stable outside of a membrane. So yeah, there's lots of things. We'll get into why we were willing to include those as targets, even though we couldn't evaluate them in our in vitro assay later. Great. So just to give you a sense of what these things look like, this is a set of 8 targets. And we're just showing you a GIF here of all of the different VHHs that we've designed to bind to these 8 different targets. There's a lot of targets, so you can see here in green and blue are different crops. So things that are green are the portions of the structures that are being cropped out of that target. And what we're actually using here are the AFDB, so the AlphaFold database structures. We'll get into why, mostly for simplicity, right? We don't have to sort of parse a lot of PDB crystal structures of varying quality, but also because those structures come with PAE matrices that are useful for truncation, which we'll talk about later. But yeah, a large part of what we're trying to show you here is that these receptors, these cell surface proteins, are not sampled from any particular distribution. We're not. They're just things that we sample from the proteome because they were of therapeutic interest to us. We thought we could design drugs against them. So we get all kinds of different domains and structures, and there's a lot of them. And this isn't even all of them. This is I think it's most of the targets now, and a very small subset of the actual, the million plus binders that we're actually evaluating. Yes.
What does it mean that different poses have different parts of the target truncated?
Yeah. So we're actually trying to design to different epitopes on each target. So really, the number of actual unique targets is much larger than just the 400, right? If you think about every different surface on that protein is a different target that we're designing against.
And was the sample to be like biologically interesting, or have you just sort of randomly cut it into?
Yeah, we used a solvent accessible surface area threshold. So essentially, we just looked at the surface of the protein and said, please choose random spots on the surface of the protein where it is solvent accessible and try and design there. So it is not. It is. Yeah,
but it's not. This paper says hit here to activate.
Exactly. No. So we wouldn't have been able to do that, or I would not have done that on 100 or 400 targets, right?
Yeah.
But so many of these targets are just. I don't want to say they're unknown to science, but they're things that people haven't really. There aren't structures of these in the PDB. There's no known binding partners. These sorts of things. They're.
Oh, I see. So
yeah.
These are not previously considered to be therapeutically interesting targets. They're just like whatever's there.
Exactly.
Some of them are.
Many of them are. I mean, it's funny. The human proteome, you realize it's only 20,000 genes. So a lot of them, have been very well studied, a large portion. But yeah. Yes.
So earlier you talked about killing uncorrupted trajectories before they finish.
Yes.
Have you looked at which epitopes produce maybe too many of these and which ones are generally producing productive trajectories?
That is also an ongoing area of research. Yes. The question was, are there, have we seen what makes an epitope unproductive versus productive? Yes. Yeah, there's definitely. There are rules of thumb, like things that, it's all of the things you might expect, right? It's strong secondary structures, exposed, like hydrophobic, large aromatics, these sorts of things make for good epitopes, especially phenylalanines. Like if you've got a phenylalanine sticking out of your protein, you're gonna get a good design. Yeah. Yes, sir.
So the region, is that the region that's like the hotspot, or is this like a region that you actually
Yeah, that's just the region that has been cropped and is now being presented to mBER. Green is everything that's been removed. Yeah, we can talk about the cropping algorithm a little bit more later. Yes.
Do you like filter after to make sure like it doesn't go into green region, or?
No, but we should have. Yes. It does. It can do that. Yes. It mostly doesn't, and I have a theory as to why this is the case, which is that AlphaFold Multimer was trained on cropped structures, and so we think that when we are doing the cropping, we are doing not just a sort of a positive, selection as to where it's not just a carrot of the hotspot of saying here's where you should go, but that every sort of chain break you make and every crop and weird non-physical thing that results is actually sort of a stick that tells AlphaFold Multimer not to put something there. Is my pet theory, but yeah.
How do you avoid finding that?
Yeah. So again, we think we were very concerned about this initially when we were doing these crops. We think essentially AlphaFold Multimer recognizes that that's not like a real thing. In the training data, whenever it's seen a weird exposed hydrophobic core before, that was probably a cropping artifact, right? In the same way that it is in our data set. So, yes.
Have you ever considered when you're making these targets or doing the testing to have like a mutation on the surface of the area where you might want it to bind? So that way you both a positive and a negative selection for these proteins.
That'd
be introducing like a known binding partner, some competitor in there or something like that. It seems like that would be a natural occurrence here if you knew what site you wanted to.
Yeah, that'd be really cool. I think it absolutely makes sense to do that. You can even use like protein MPNN to redesign surfaces so that they still fold the same way but don't have the same, they're still functional. I think that'd be a really great avenue. For our purposes, we just aren't interested in any particular epitope that much. And so, yeah. Great. This is sort of the big experimental results slide. It is maybe hard to read, so I will walk us through it carefully. What we're showing you is this heat map that has all of the results. Every row in this heat map represents all of the binders, all of the VHHs that were designed against a given target. So every row is a different target and all of the binders that were designed against it. Every column is that target, those wells in the experiment. So this is that all versus all matrix of sort of binders to targets. But really what we're showing you is we're compressing all of the binders to a given target into one row so that we can show you the rate of success. So a dark blue here color represents a rate of success of about 5%. So it means that 5% of these binders are binding to that target. We'll get into how we're doing that hit calling in a moment. But so everything on the central diagonal here represents on-target designs. It represents things that were designed to bind to a target that we are seeing specific binding to that target. So what you'll notice is that we do see, and this is sorted by on-design success rate across the different experiments. So we are seeing, significant elevation in on-design success rate. What you'll also see is that there's a number of targets, like in the bottom right here, where we do not see any on-design success rate. So these are targets for which we are failing to have any binders successfully bind. And then the other thing is that we have things sort of off the diagonal here, where this is now represents what we call off-design hits. So these are binders that we designed to bind to one target, but we are seeing them bind to a different target in a specific way. So we're only evaluating 145 of the 436 targets here. And yeah, it's this all versus all hit calling. We'll talk in a little bit about how we're actually calling something a hit or not a hit. But yes, this is the sort of overall results. We're able to successfully get hits to about, what is it?
Like
45% of targets in a way that is elevated over that off-design background. Yeah.
Cool.
So 45% of that 145. Yes. Yeah, so we found that there were some targets that were sort of more promiscuous than others in a specific way, right? It's interesting. They're just it's very possible that they're have a propensity to bind just sort of the framework, regardless of CDR, for instance, might be an explanation. Yeah. Yes.
There are proteins like chaperone that have 5 months of stuff, so you have to pick something that has some approach. Exactly.
Yes.
Finding for any of these target designs and doesn't predict that they actually bind.
We really want to be able to do that. Part of the difficulty is that this, as much compute as it costs to do just the diagonal here, imagine trying to do now the entire matrix or a significant portion of it. But the real answer for why we haven't really done this is because AlphaFold we think is not that good at it. Ultimately, whenever we try and say like, hey, here's some antibody sequence, we don't know where it binds, and ask AlphaFold to fold it. At least AlphaFold 2 doesn't do it particularly well. So that we think that problem of it's a funny asymmetry where we believe we can design something, and if we then test it, we can be confident in where it binds. But if we didn't design something, if it just is occurred naturally or came from somewhere else, or it's binding in some other way that we don't expect, it's binding some new targets, we have no prediction of its epitope, that we can't really predict its epitope, at least confidently.
Yeah, it's like AlphaFold can only fold certain antibodies, but if you ask it to design something, it's only going to design the things that it can fold, right? So you still would fail like naively on like 80% of folds, but all the designs are things that it did think were good, right? Cool. Should we? We're running up on time here, and do we usually go over or okay? We'll try not to keep you guys too long. I will.
Don't rush. You can take it.
Okay.
I feel bad if anyone asks.
That's why we serve here.
Yeah. Okay. We promised we'd talk to you a bit about how we're actually calling hit from our continuous NGS phage data. This is the answer. So for each target design pair, well, really for each target, we have 4 replicate wells. And every single sequence in the library is tested in every single one of those wells. So some signal that you might expect if you have, sequence 1 that binds target A, this is abstract representation, but you might see a mini bit of the heat map that looks like this, where you see, oh, look, there's signal in all 4 wells. Maybe it's a binder, right? But just alone, from a phage experiment, you wouldn't actually be able to distinguish this from, really high background. There's a whole bunch of other problems that could pop up here. So we make a few key assumptions, really one key assumption here, which is that for each one of these replicates, we assume it's sort of like identically, identically distributed, whatever. Is pulled from this distribution of how much bound phage is there, conditioned on like the actual interaction physics, right? Like what we want to get at is like, do these things actually bind? What we would like to have is the number of bound phage. What we actually have is the like NGS counts of the sequences, which is like many layers removed from that, right? And it turns out like modeling F is messy, and you shouldn't attempt it. Or maybe you should, but like we don't in this case because we have an easier solution. So zooming out a bit, if you looked at this matrix of like all the targets and all the sequences and what the signal looks like, you might see something that looks like this by eye, where you'd look at this and you go, oh, sequence 1 probably binds target A, sequence 2 probably binds target B, maybe sequence 600,000 doesn't bind anything. And like how do you distill this into a single metric for hit calling? Well, one way, and I bolded here, the power of replicates and order statistics. One way is like, imagine just comparing to every other well. Like we have 145 targets, this across 2 experiments, but it's like 512 wells essentially. And that includes some empty wells as well. The strictest thing you might be able to say is like, sequence X is a hit to target Y if and only if signal across every replicate of target Y. And I probably should have used the letters here for clarity. But basically, like imagine that the signal here across every one of these 4 replicates is greater than it is for every single other well where you're measuring that sequence. Well, if that's the case, the thing to think about here is like, well, what if I like randomly drew 4 of 512 ordered items? How likely would I be to draw like all of them as the top 4 items, right? And from this, we can essentially assign a false discovery rate. It turns out that if you have 512 wells and 4 replicates per well, that false discovery rate is 10 to the minus 10, which is quite small, even in the context of 100 million interactions. There's a weakness to this, which is that, well, for one, it's super strict, and for 2, it doesn't allow you to measure things like cross-reactivity, where you might see multiple targets with high signal, right? So we do want to account for that. Yes.
By signal?
Yeah. So
essentially just counts. Yes.
Yes.
You can imagine being normalized by well, for instance, or I think we use spiking controls, for instance.
Yes.
That's my, it's like, how do you normalize?
Yes.
Is the thing.
Yes. Yeah.
Yep.
So we use a spiking control to normalize.
Yeah.
That just makes every well comparable to every other well.
Cool. So what you might see instead for some sequences is some degree of cross-reactivity. And as Mike alluded to earlier, there's actually a difference that we draw between like polyreactivity and pan-reactivity, which is important when you have a lot of targets, especially since these targets were selected, like some of them have homology, right? Like some of them are quite similar to each other. So we might expect some designs to bind multiple targets, and that's actually not a failure. That's still a success. So we want to allow for, some leniency here. It's a simple change here. We just say, sequence X binds target Y if the signal is greater than like some number of other wells, and you can move that number up or down to adjust your false discovery rate. In practice, we wanted our false discovery rate to be like approximately, 1 per target. So we chose to make it, higher than all but 20 wells. This also allows for 5-fold cross-reactivity at most, since there are 4 replicates per target. Yeah, like I said, a false discovery rate of 1 in a million is not that bad. When you have a million designs, 145 targets, that's you call 1 hit to every target. That's wrong. It's not a big problem for us. We have a lot of hits. So with this in mind, we can look at just a couple other patterns that you might see, and some of which Mike has already described. So of course we have our, like, imagine you design a binder against target A. That's what we're looking at, in the picture on the left. You might see, yes, indeed, we hit target A. You might instead see we didn't hit target A at all, but we specifically hit target B, which is an interesting thing to happen. You might see I hit both target A and B, or maybe both target B and C and not A. So this is this sort of like low degree polyreactivity that we're talking about. And importantly for a hit calling algorithm, like pretty much everything else is not a hit, right? If there's inconsistent or low signal for any target, like if any well is 0, it's never going to be called a hit, obviously. If every well across everything is high, it's not going to be called a hit. So we completely disregard things that are pan-reactive and sticky. We're only paying attention to on design, off design, and sort of low end polyreactivity. Yes.
The last rule, which is pan-reactive, like how much because
how many of our sequences
is going to be wasting? It's going to occupy all of your sequences.
Yeah, how many of our sequences were pan-reactive? That's the question. That's a good question. I don't think we've formally done. Have we done that analysis?
No, it's a small fraction. I'm just looking at large heat maps. It's less than 1%, but they're there. Yeah.
Yeah, a lot of this comes from like, we developed the hit calling algorithm by first eyeballing a heat map. We love staring at heat maps, and you can learn a lot from that, and then you can develop things like this. So yeah, I mean, from the eyeball analysis, staring at the heat map, it's not a whole lot. You do see it sometimes. Okay, we can move fast through this. This is just sort of pointing out, so like what is on design hit rate, what is off design hit rate, and what is actually a different concept here, which I think is more common, which is off target hit rate. So, the first thing is like, yeah, if we design against target A, we hit against target A, we want to know out of the designs that were both designed and hit target A, out of the designs against target A, how many both hit and were designed? That's on design hit rate. We have a separate concept of off design hit rate, which is literally anything else in the library, which we sort of take to be like a naive library, from our design method, but they have no information about the target at hand. We use that to compute an off-design hit rate. And then there's like another concept here, which is like, well, if I design against target A, how often does it hit things that aren't target A? And we kind of argue it's not that important for our analysis here. It doesn't tell you how good you are at designing against target A. It maybe tells you how bad you are at designing specific things. But you can also get that by just our hit calling algorithm. So we largely ignore the third box here, and we pay attention to the difference between on design and off design hit rate. And here's a plot showing for all targets where we had, I believe this is all targets where we had any on design hits, could be significant on design hits, what those hit rates were, subject to various IPTM filters. So apologies for the low resolution legend here. I screenshot it from the paper this afternoon. But the 3 bars here for each target are all the binders we designed with no filtering whatsoever, just raw mBER designs. Then what happens when we apply an IPM filter of 0.7, a subsequent filter of 0.8, and then in the red line, these are the off-design hit rates. And we make the argument that if your on-design hit rate is greater than your off-design hit rate, the off-design hit rate is like the rate at which you might just accidentally hit this thing. Well, you can quite simply just do a statistical test there. You can do the Fisher's exact test, and you can determine whether you are significantly, having design success against the target of interest. So what we see here in the pie chart is for 53 of our 145 targets, we have a high significance on design hit rate. This is p less than 0.05 divided by the number of targets, the Bonferroni correction, which is a little more stringent than it needs to be, which is why we include the other boxes here. So we have medium significance, which is p less than 0.05, maybe not stringent enough, and then low significance, things that we had specific on design hits, but it wasn't significantly elevated over our off design hit rates. What we see here in the targets is a little bit easier to see on the next slide, which is that, yes, as we increase our IPTM filters, we do in fact see much higher hit rates. Going from no filter to greater than 0.8 here is like almost a 10, maybe it's not quite tenfold, but it's getting there. And we do see for the highest level filter here that for the lucky special nicely behaved targets, we have hit rates ranging into like the 30 to 40% range, which is quite good for VHH design. On the right here, we're showing the distribution of IPTM across the 2 libraries. So there was one main difference between the design, well, there are really 2 main differences between the design of the libraries. Library 1 was against a smaller target set, and we also sampled across the range of all possible IPTMs. We want to learn about things that were poor predictions and also things that were good predictions. Library 2 happened in a later experiment, and we said, let's just take things that look good. And you can see in both cases, there's a correlation between on-design hit rates and higher IPTM. Maybe not as high as what we saw earlier with RF diffusion, but still, respectable that we can get a roughly 4%. Maybe there's some different experimental conditions here. Maybe we undersampled the library. Still higher hit rates with higher IPTM cutoffs. And importantly, that IPTM does not really correlate with off-design hit rates. So we're not like we don't get better at designing at things that we didn't mean to design against by incrementing this filter, right?
Yeah, so this is another signal that we think is indicative of true on-design success. We wouldn't see this pattern with IPTM if we were just randomly succeeding through the law of large libraries.
Yeah.
Yes.
Did you ever go back and apply Ron Dunbar's IPSE analysis and see if that goes higher than is more predictive than the IPTM and your analysis?
We would love to do that. We did not save the PAE matrices because they are really big, but we're working on it. We're going to refold them all. So we'll let you guys know. Yeah. Great. Okay. Quick aside here, just because I thought people would be interested. How does mBER compare to diffusion-based methods? So earlier before mBER, there was RF antibody. Maybe a month or two after mBER, Boltzgen came out, which has been really exciting for the community. We want to give a quick, we haven't had the chance to test these head-to-head in vitro, but we have some in silico comparisons for you. Here is mBER versus RF antibody. This figure's in the paper. On the top, we're looking at the distribution of IPTMs that both of them spit out when you fold the design against the target. As you can see, RF antibody designs very few successful things versus mBER has quite a few, a decent amount of designs, in this lowly sampled set of targets, right? And if we were to look at the sort of pass rate at 0.7 IPTM, which is a common cutoff for us, RF antibody has 14 of almost 10,000 designs that pass that, whereas mBER has around 16,000. And this benchmark was run against 5 different targets. So here you can see the targets, and for almost all of them, the max IPTM score, which again is our main filtering metric, is much better for mBER. We decided to run a further comparison. This is actually a library that we have just designed. It is a set of binders to 52 novel targets, specifically for manifold drug programs. So we can't share the target names with you, but we can share the in silico success rate. So these are all targets, many of which don't have any known binders, which we are very interested in therapeutically. We ran mBER and Boltzgen both for the same number of GPU hours. And these are the number of passing designs from each, with mBER in blue and Boltzgen in I guess magenta here. I want to caveat here to say that this is Boltzgen in our hands from a first try, and mBER in our hands from having worked with it for over a year. So I don't want to present these results as Boltzgen is bad. Boltzgen is a very exciting method. They do a lot of really interesting things, and they have some really cool successes. This is just what happened when we ran it in our own production experiment. We can look at a couple other views here. This is the IPTM distribution. So as you can see, actually this is the internal version of mBER. We hope to propagate some of these updates, mostly in weights and stuff, or loss weights to the public version, but we do a bit better in generating high IPTM stuff, whereas again, Boltzgen is quite low. And we can also look at what I think is one of the most important metrics for design algorithms, which is how long does it take you to get one success? Where again, in our case, success is in silico and it's greater than 0.7 IPTM. But what we've shown here is that, again, in our hands against these targets, we achieved one success every roughly 2 minutes of running mBER, with very high variability across targets, to be fair. Whereas with Boltzgen, it took closer to 12 minutes per success. So in our hands, again, mBER has been much more efficient at generating successes. We can also look at diversity of the things that we generate. So this is just sampling some set of the sequences and comparing the sequence distance, Levenstein distance. And this is not you'd have to do n-squared comparisons to do the whole thing. This is some sampling, but the sort of median distance here for this set is roughly 30 amino acids different between 2 different mBER designs. They're quite diverse. So is Boltzgen. They're actually both quite diverse again in this experiment.
gen versus M? The same amount of GPU hours, how many models? Yeah.
How many total designs? I had a plot of that and I didn't include it. Boltzgen generates a few more. I think Boltzgen is a little faster per design than mBER. This is especially true. I won't go in depth on this, but mBER, I'm showing the sequence diversity across different mBER trajectories. In one trajectory, oftentimes what we'll do is we'll actually sample 10 sequences that are like not that far apart, because that helps us sort of experimentally, like it's sort of like doing a very small mutational scan. So
It's also more computationally efficient to do that.
yeah. So
mBER is more complicated. With the
yes, mBER, we are in our hands in this experiment, mBER was more computationally efficient than Boltzgen. Yes. Yes.
Like Boltzgen designs, did you ever check how confident the Boltz refolding model is with your ML designs?
Not yet. Yeah, it's in the works. We're standing up a bunch of evaluation pipelines to answer all those sorts of questions. Cool. I'll pass it back to Mike.
Great. So we had some questions earlier about hotspots and epitopes. So we'll just go into briefly how we did that. Hotspots are very important because for a variety of reasons. The first is that you might actually care about the epitope you're binding. For lots of therapeutic applications, you want to bind to a specific epitope in order to block something, or maybe to avoid blocking something. So being able to control that is very useful. But actually, there's a lot of practical reasons to pick hotspots anyway. Even if you don't care where on the target you're binding. For one thing, making sure that you are diversifying your binders as much as possible helps you sort of hedge against the risk that the structure that you think you're designing against might not actually be the real structure when you test it. So a diversity of binders diversifies your risk there. The other thing, though, is that it actually really helps computationally to have a hotspot and thereby truncate or crop your target around that in that way. So these models, their computational efficiency scales very poorly with target size. So if you try and give a full target, oftentimes it won't even fit in memory, in fact, for a lot of proteins. So being able to pick a hotspot and then crop your target to just the relevant information is very useful. Our algorithm is fairly straightforward. Basically, we just pick a hotspot, draw some sphere around it in angstroms, and say all of the residues inside we want to keep, all the residues outside we want to don't keep or not keep, and we say give it some penalty for making a new chain break, and then sort of find the optimum given that criteria of which residues it should include and which it should exclude. The other thing we do is we're using a lot of AlphaFold DB structures. Part of that is convenience. Part of that is because those structures come with PAE matrices, which we can use to understand whether or not, AlphaFold will often fold things back on themselves. So, there's lots of proteins in the proteome that are very elongated. AlphaFold likes to fold them back on themselves. But those two ends might not actually be next to each other in a meaningful way, right? But that they would sort of sterically block each other in terms of in the design space. So we can use the PA matrix to also further filter and truncate and only keep the most relevant context for the model. So we're just going to zoom in on some examples here of successes for mBER. This is a transferrin receptor. What we're doing here is we're highlighting all of the hotspot residues that we chose as a colored residue on this surface. So they'll be red or green. Red just indicates that we did not get success to that hotspot. Green indicates that we did. And we're showing the structures then of those hotspots where we did see success. So TFR, we see some success, but not too much. FcRn is another therapeutic therapeutically relevant target that we're designing to. Here, we do see a lot more success. Importantly, we are still only seeing success to roughly half of the hotspots we chose to design against. That's going to be a continuous. That's true for things generally. The majority of hotspots across all of the targets we looked at, we just don't see success against. And so when you're looking at 1% success rate to a target, it's often even, it might be 10% success rate to a single hotspot on that target, for instance. And so again, we yeah, more or less. There's lots of other things we can talk about here in terms of IPTM success rates. We see those IPTMs increase our success rates. And then PDL1, we thought we maybe would have found some PDL1 binders that didn't just bind to the classic interface of PDL1, but in fact we did not. We were just fooled by the fact that we hotspotted over here, and we actually got designs that bound prediction structures onto the other face. So beware of that. Yeah. And this is an example. We wanted to sort of make the point that the targets we're going after here are not canonical design targets, and we actually of those 45% of targets that we successfully designed against, 16 of those are actually targets that don't have any sort of known structurally defined interaction in the PDB. So these are things that there are no known, or at least no crystallized structures for, and so there can't possibly be any sort of contamination in that way. Okay, the other question one could ask here is, what are we doing with all these binders? Why did we do this? We mentioned that we designed against 436 different targets, and we only tested against 145. That seems very curious, right? And the answer is, what Manifold's real, we're doing a whole lot of in vitro multiplexing, but really what we're trying to do is in vivo multiplexing. So we have developed a suite of technologies that let us test molecules at scale in living organisms. And so that's useful because we can understand sort of what the living organism is doing to those molecules in terms of their pharmacokinetics, what's happening, whether they're being degraded, whether they're where they're going in the blood and how long they're lasting, as well as which tissues they're being driven towards. So we're very interested in understanding and developing methods for delivering therapeutic payloads to specific tissues. So essentially, unlocking delivery of things to the brain or the kidney or the heart. And so we're interested in designing binders to 436 targets, even though we couldn't screen the majority of those in vitro, because we can screen them in vivo. So I'm just going to give you sort of a sneak peek of some of this data. We have this multiplex in vivo readout where what I'm showing you is a heat map of signal in these different tissues. So this is done in non-human primates, where we've taken thousands of designs. These are designs to a single target, and screen them for their propensity to go to these different organs in the animal. So you can see as I'm sorting by brain enrichment, it's just an example of finding a bunch of binders to a given target that show a biodistribution profile of very high therapeutic interest. And of course, biology is messy. It doesn't all look like that. This is a subset of binders that were all evaluated in a single experiment in non-human primates that are going to a variety of different tissues. So you can find things that enrich specifically in adipocytes or in spleen or in the kidney and the pancreas. So it's a large, exciting data set where we're sort of leveraging the ability to use AI to design against so many targets that even though we can't test them in vitro, we can evaluate them in vivo and identify novel targets with novel potential for delivering therapeutics and medicines. Yeah. Yes. The technology here is proprietary, so I can't talk about it too much, unfortunately. Yes, they are VHHs. They're VHHs. Of course, of course. So we actually have several different technologies in this space. I'm showing you what we're calling ultra high throughput vivo. We have, and it's a very high throughput modality where we are able to understand tissue biodistribution. It's less useful perhaps for understanding pharmacokinetics, though it still has some of that data. We have other modalities we call high throughput vivo that are still at the scale of, say, 100 plex. So we can test 100 molecules at once in a single animal, where we do get really nice pharmacokinetic data that we study. Exactly. So we can extend their half-life in that way, yeah. And our FcRn binders are interesting in that sense as well. Yes.
And you are doing 100 molecules per animal?
So in this study, it's actually tens of thousands. But yes, we do regularly do studies at the scale of hundreds of molecules per animal. Great. So this is the engine manifold building. Just as a small pitch for the company, we are using AI guided design to really mostly just to fill the massive experimental throughput that we have as a company, both in vitro and in vivo, where we can do these massive library scale screens. And the other power then is that for every single AI guided design, you have a structural hypothesis, right? These things are very valuable because they're not just a naive binder where maybe you know a target, but you don't know an epitope. You don't know these things like the structure. It's hard to reason about why one binder might be working and one binder might not. What we're able to do is sort of label these massive structural sequence, design data sets at scale, right? So we're able to give them labels about in vitro success. We're able to give them labels about specificity. We're able to give them labels about their pharmacokinetics and where their biodistribution and where they're going and whether they're functional in a living organism. And the end goal is to take that back and make even better AI design methods, right? So train on this, learn on this, and close the loop.
Yeah, and with that, we want to point out, mBER is available open source under an MIT license. Maybe this flew under the radar for some people, but actually, to my knowledge, it is one of 3 VHH/antibody design models that is fully commercially available, being ARF antibody, mBER, and VoltGen. I believe Germinal has some non-commercial issues. People could correct me there. So if you want to design VHH and you are at a company or not, please use mBER. We're very open to feedback. Leave open an issue on
Erik will respond to the issues on GitHub.
It might take a couple weeks if I'm doing it.
I promise he'll respond.
I'll respond. But yeah, we would love for people to use it and get back to us because we open source mBER because the open source community is what got us here, right? It's we really believe that protein design is driven by this community, and that a rising tide lifts all boats here. So we want to sort of get back where we can, and we would love to incorporate also ideas from you guys that helps us make better drugs, better designs.
Yeah, we want to point out, even if you don't care about antibodies and VHHs, this same sort of templating and conditioning approach works for any format you want. So you can use mBER. We actually built it and designed it in that way to take into account, different types of templates, different types of conditioning to make a format, whatever format binder your heart desires.
Yeah, and please make new protocols, and if you want to make things that are not VHH, open source them as well. Make a pull request. We'll happily check it out, and we'd love to keep growing. Also, we are currently preparing a public data release. So a lot of people have asked, hey, the really cool thing about mBER is the data. The model's great and all, but can we just have the data, please? And the answer that we have to give is well, a lot of the data is proprietary targets, so we can't share that. But we're currently working on what we think will be a large subset of the data that is against targets that we are not going after the company, that we will be able to share to the community. Sequences, predicted structures, design metrics, and experimental data for all those targets and binders. So, yeah, expect something hopefully within the next month. And again, yeah, we're really excited for people to build on this and make even better design models, understand what's going well, what's going wrong. Yeah. And with that, I think, obviously, big thank you to the Manifold team. Mike and I are standing up here, but we are supported by a team of now, what, 50, 60?
I want to say 60. Yeah.
Yeah, we've grown a lot in the last couple of months.
This whole, yeah.
Yeah. And special thanks to our co-authors, Cypriya, who's, been in the lab and just hammering out these experiments day and night. And Pierce, our CTO and co-author, who has also been really involved in the development of this work. And there's another development slide actually, which is for you guys, because again, we really, mBER is based on all the open source tools. Notably, we did not train a model here. We're not coming out here and saying, oh, we trained like a new thing with all our GPUs. We just took, stuff that already existed and we said, can it work for our problem? And yes, it did. So thank you guys for, everything that, you've built, and we hope to continue building with the community. Oh yeah. We're hiring. We're hiring too. So if anybody's really interested in this data, we have not a lot of people actively working on analyzing it. And we have honestly more data than we know what to do with. So yeah, reach out. Yes.
Awesome talk, and thank you for open sourcing the code and for sharing the data. That's really spectacular. The data are very exciting, but I'm sure it's a lot, as you guys know, and I'm sure a lot of maybe the old school protein engineers in the audience are thinking, when you take a protein from a display format to purifying it, things can behave very differently. So just wondering if you've taken any subset of these designs from your phage display assays and actually tried to take them one at a time, and how well did they behave? How well does the single purified protein correlate with what you see on phage?
Yeah, I don't know if we have. So the question is, when you take it off phage, how well does it work? I don't know if I have like a single number for you. They do work. We have taken them off phage in fairly large number. For the for the vivo experiments, we have to, right? I don't have a good number for you. I want to say it's like more than 50%. But there are absolutely failures, of course.
I think something to note here, especially with respect to the VHH format, is we're fixing the framework sequences, which are largely responsible for stability of the VHH. Of course, the CDRs can cause problems, but that's why we use the language model to do some biasing here. So unlike other design models, pretty much every other, there are probably some exceptions, but pretty much every other design model out there uses protein MPNN or some sort of inverse folding step to do redesign. We're using known framework sequences, right? So 80% of the things that we're generating are stable sequences already. It's really just the loops that we're designing.
Yeah.
Sure. Yes.
So continuing from Chris' question, we've been able to, for some of the success on heats, been able to purify, find KDs, or even crystallize or structures.
We have not crystallized anything. So the question is, how much more validation have we done here? We do have KDs for a number of them. They're sort of maybe what you'd expect, ranging around 100 nanomolar. That's sort of a little bit better, a little bit worse. That's a typical range for us.
And no structure.
We have not crystallized anything. Yes, in the back.
Well, have you done any experiments with heavily post-translational modification targets or proteins themselves? What does that look like?
Yeah, it's something that we haven't paid as much attention to as we would like to, as a result of the large number of targets we're often going after. We do think that is a source of failure. Actually, so oftentimes when you buy a recombinant protein, it will have been produced in a mammalian system, and so we'll have post-translational modifications, which, since we're using AlphaFold 2, which doesn't even support the ability to take into account those modifications. And we're also not, when we're doing, you can imagine when you do epitope selection, you could try to avoid those regions, that sort of thing. We're not doing that. So that's absolutely a failure mode that we are sort of blind to, but we know it exists. Yeah, we didn't make the problem easy for ourselves.
I think we have a lot of questions in the chat. I think I would love to get everybody, but I feel like we've been ignoring these guys for a while. So maybe we can quickly go through and see if there's anything. to answer real quick. Do you also use PLM embeddings for the target as well to evaluate the relationship between target and CDR with using both the CDR only and target only embeddings? Target as well. We do not use PLM embeddings on the target presently. It's just for the binder. Do you ever test your total pool against a polyspecific? Well, no, we don't test against polyspecificity reagents because we have a lot of them. It's all the other targets. How long does it take to complete the wet lab validation, including the whole phage library creation and NGS readouts? 2 to 4 weeks. Is that fair to say? Is it possible the binders would have clashes since you used crop targets when running the binding assay? Was it the full proteins or crop proteins? We answered that. Yeah, we should pay more attention to that. It is possible.
In fact, sometimes when you find a really high IPTM things, like if I see an IPTM of 0.93, oftentimes it is because it is binding where the crop target used to be, like where the crop protein was, the binder has now taken its place. And AlphaFold says, yes, that looks correct. That's what there should be protein there.
Why did you not use TightSeq to also get the KDs through sequencing?
I don't remember. I have heard of TightSeq. I don't remember it well enough to talk about it here. So I plead ignorance.
But the paper is shared in the chat if anybody's interested. Did you test using versus not using protein-MPNN? Why did you not choose to use protein-MPNN? We already sort of answered that. The frameworks are already designed, and we wanted to keep the interactions AlphaFold designed. So no protein-MPNN for us. Do you also use... Okay, we already... Yes. Okay, great. We've got the chat. Any other questions? Yes, to the front.
Yeah. I have a question about the polyspecificity. It seems like when you process your data, you do your calling, you have 3 labels per design, right? Either it's on, off, or polyspecific.
Well, for each, how to put it? Really, what we mostly report is just on design success and off design success. Whether something is polyreactive, we're not, at least in the results we're showing, we're not like specifically labeling that. So we're just showing, success rates.
Okay. I guess
those labels. Yeah, go on.
Sorry, it is kind of half-baked, but I'm wondering, like, what can we learn from that polyspecificity?
Yeah.
Specificity information, like, how can we use that in your next generation?
We think it's really useful. We think we can learn on essentially polyspecificity. What it probably represents are, epitopes or paratopes, really. Those pairings that are sort of promiscuous or common, maybe, right? So you can imagine some binding interactions are going to be very specific as a result of sort of good shape complementarity, or at least strange shape complementarity, right? Shape complementarity that's not very common among the other epitopes that we're testing against. There's also all sorts of other signals for sort of polyreactivity, like a hydrophobic core of the binding interaction might represent something that tends to be likely to bind multiple targets. It's an open area of research. We're very interested in trying to use this data to learn that and make the design algorithm better.
Briefly, one analysis we tried to do is we said, well, maybe a lot of the polyspecificity is being driven by homology or structural homology between targets. So we did a first pass at some sequence alignments and some like fold-seq runs just to see if that similarity was correlated with polyspecificity. We found that for a few targets that was the case. There were a few pretty obvious examples where 2 targets were very closely related, and we saw binders reactive to both. But it didn't explain a lot of it. So still more analysis to be done there. Yeah.
Yes, on the left.
This time to design.
Yes.
Basically curious about the difference in variability presumption. So the N and the green of the event is the same. Checking the same number, the same targets.
That's correct. Yes.
So what accounts for the much greater variability?
There's 2 or 3 effects here. One of them is that Boltzgen just wasn't successful against a lot of the targets. Another effect is that let's see, for mBER, I guess there are cases where you just get a pathological success here and there. I wouldn't even I wouldn't even be confident that like these guys have any real successes in the sense that like this might be where we're tricking the model and getting like 0.9 IPDM from like black holing everything and all the atoms are like close together or something. But also sometimes it happens that like, for a really big target, if our crop is really big, the algorithm takes longer to run. If that happens to also be a target that we only get in silico hits 1 in 100 times, then that'll show up at the top there. So yeah, I can't speak very well to like why Boltzgen doesn't have stuff up there, but my best guess is that it's just because there weren't a whole lot. There were a lot of targets where it had no successes that are just not being shown.
Yes.
You mentioned just a couple frameworks. I'm curious how you pick those, and then if you notice whether some of them might be more prone to off-target effects than others.
It's a great question. We designed the frameworks that we use in mBER before designing mBER. It was actually part of the antibody-specific fine-tuning work we were doing. We designed them based on like the human germline IGHV 323, which is a pretty common one for humanization. And in the this is getting the nitty gritty, but for people who are really familiar with VHH, in the framework 2, there are some like hallmark mutations which are really important for like CDR3 conformation. So we basically sampled from the set of known antibody sequences the most common like 10 sets of hallmark mutations. So yeah, we designed everything else based on IGHV 323, sampled the most common hallmark mutations, and then just applied various CDR lengths. We do see variation in success across different frameworks. I don't have a plot for you, but there are differences. And there are also differences on a per target basis. Some targets prefer certain frameworks over others. Yeah. Yes.
Yeah, so in other Minecraft type methods I've seen, they sort of suggest that you might have to tune the weights of the output loss a fair amount, depending on target. And I'm guessing, like, well, I think you did it based on what you said, right?
Yeah.
But like, how much do you think you might be like leaving on the table?
Probably a lot. Probably a lot.
Absolutely.
Yes. Yeah. I mean, for what it's worth, we against like reasonably well-behaved targets, we do see like roughly 20% in silico success rates. So our efficiency is quite good. It might be the case that for like hard targets, tuning would be really helpful. It also might be the case that for easy targets, tuning would get us close to 80% success rates. We're working on solving those problems. So stay tuned, I guess. Yeah. Yes. Yeah.
Really like the discussion of how exactly you converted the NGS reads into physical 1-0 hit or no hit output. I'm wondering if you considered, rather than interpreting it as a binary cutoff, as more traditional claims to spectrum, because I imagine that there's probably some sort of relationship between the affinity of the binder and the number of NGS reads that got out of the binder. Yeah,
there's a lot of ways to do that analysis. The short answer is that like we have done comparisons between NGS readout and like affinity in other experiments. Oftentimes, what you'll see is that like above a certain signal cutoff, you get something that's binding, and then like past that, there's not a whole lot of correlation between like NGS signal and affinity. There are exceptions to that rule, like there are things where you see like really, really high signal and they're really, really tight binders. But it's not good enough for us to use that purely as like a affinity measurement, and so we stray away from that. We'd rather use well-defined sort of statistical hit calling. Yeah.
Yes, Sergey.
I'll ask one. Maybe more questions after this, but we should probably wrap up at some point. But essentially, in one of the slides you mentioned there's like 15 or so targets that were not in the PDB.
Yeah.
I was wondering if you could say a little bit more about how many of the targets that worked had antibodies that bound to them versus the ones that didn't have, like,
is there a bias
there?
there?
Trained on antibodies. That makes me wonder how much of it is recapitulating things that maybe are in PDB versus not.
I don't think we have a number for you. Just to say that we have one interesting caveat is like when you tried to design one of the targets, for instance, we didn't get success to was HER2 because I don't know, we just can't, like, that is a very well understood, there are known antibodies that bind there. We're not able to get a binder even though we're hotspotting in the same for trastuzumab or whatever. We don't think that that's what's happening. Maybe we should do a more systematic exploration of all of the known antibody binding sites. Sorry, I don't have a satisfactory answer.
No worries. I mean, I think there's a few AlphaFold 3 predictions that requires like many rounds to make a good prediction.
Mhm.
And there's some of them that just predict like the first round. Just using prediction always predicts the right sites.
Mhm.
I wonder if there's something special about the sites that makes your approach work for those versus other targets. Might be interesting to sort of analyze the sites where the binder goes and whether or not there's something special about the ones that work but don't work.
Yeah, all I can say is, oftentimes we get asked, why can't we just make a binder that binds the place where we know a VHH binds, right? Here's a target, here's a VHH, here's a crystal structure, right? Make this, and it doesn't work. So I don't know, it doesn't seem like it's that simple, or it's not a one-to-one in that way. Whether all of our design, there's lots of targets for which there are no known structures, and so those must be unique. I guess we're just not even concerned enough about this problem that we have explored it. That's frankly the answer.
Yeah, another big thing is, if we think about like the truncation really does a lot of heavy lifting, I think, where, if you feed AlphaFold the whole target, like maybe it's liable to say, I've seen this target before, like I'll put it in the usual place. But if you give it just a truncation of the target, even if it's a target that AlphaFold's seen before, like maybe, that surface is like, close to the other things AlphaFold has seen, but not on that target. And so, you get like generalization maybe for free in some sense by just, hiding the stuff that you don't want it to overfit on, right?
Well, let's thank Mike and Erik one more time for. And just a quick note, so next month we're going to be back here at MIT. We're not alternating the locations. And we've got a real treat. We've got Jeff Gray from Johns Hopkins is in town, and we managed to get him to come over. In order to accommodate Jeff, we had to switch the schedule, so it's not the second Wednesday of the month. We're going to be here on February 25th. So see you back here February 25th.
Thanks.
And there may be more beer, so stick around and ask more questions.
Yeah, we'll stick around if people have conversations.
Shall I end the Zoom? Farewell.